Hello everyone, welcome to Sri classes. So today we are going to solve some question related to our previous lecture. So if you have already seen my previous lecture, it was all about the structure of DNA. And in case if you have not seen it, it's a small request, kindly go through it first, because today whatever question we are going to solve, it is totally completely related to that particular topic, that is all about structure of DNA. Nucleotide, what is nucleoside, what are the different bonds between the sugar, between the nucleotide, and between the nitrogenous bases. So kindly go through the previous lecture if you have not seen it yet. So let me start. As you can see, this is the first question. This one is your textbook question. Here it is given, the length of E. coli DNA is 1.36 millimeter. Can you calculate the number of base pair in E. coli? So as you can see in the question, only the length of E. coli DNA is given. And they are asking to find out the number of base pair in E. coli. अगर आपको याद होगा तो हम लोग क्या पढ़े थे कि जो DNA, what is the structure of DNA? It is a double helical structure. You can see this is the structure of DNA. It is a double helical structure. Double helical means what? Double helical means there are two strands. They are arranged. They are intertwined with each other. Okay, and they are forming a structure like a helix. Hence, it is called double helical structure. And you can see the picture itself. What the picture is showing. You can see that the DNA is taking, there are some turn in the middle. Okay, the two strands, they are intertwined with each other, and there are some turns in the middle. This one is one turn, this one is one, this one is one. So they are turning at some point. Now the question is, how many base pair are there in between these two turn? This is one, this is one. So in this one turn, how many base pair are there? We have already read about it in our previous lecture. Total number of base pair in one turn, it is 10. Total number of base pair in one turn, that is 10 base pair. And what is the distance between two base pair? When I'm talking about base pair, what does it mean? Can you see this? These are what? These are base pair. Again, when I'm saying base pair, what does it mean? Base pair means I'm talking about the nitrogenous bases. They are paired with each other. They are paired with each other. What are the nitrogenous bases? Nitrogenous bases are broadly classified into two types, purine and pyrimidine. Under purine, adenine and guanine are coming, and under pyrimidine, cytosine, uracil and thymine. So these are the bases coming under pyrimidine. So A will always pair with T and G will always pair with C. So this pairing between the bases, that is what called base pair. You can see this. A pair with T and G pair with C. So this is a base pair, this is a base pair, as they are present in pair. Hence they are called base pair. So this is one base pair, this is another base pair. Now the question is, what is the distance between these two base pair? If we look at the distance between these two base pair, you can see the distance between this and this gap. I'm talking about this gap. So the distance between these two base pair is 0.34 nanometer. If you can recall, we have already read about it. The distance between these two base pair is 0.34 nanometer. And the question is, how many base pairs are there? If someone will ask you how many base pairs are there in 1 ton, so you can easily say that there are total 10 base pair, because we have already read about it. It's a factual thing. So how many base pair are there in 1 ton? 10 base pair. What is the distance between two base pair? The distance between two base pair is 0.34 nanometer. So if someone will ask you what is the length of that particular ton, what is the length of that particular ton, you can simply calculate 0.34 into 10, that is 3.4 nanometer. So the same question, this question is entirely based on that only. You need to solve this question on that basis. Here, the length of the E. coli DNA is given. Suppose this is the E. coli DNA. Here the length is given, and you know what is the distance between two base pair? Just remember one thing. अगर question में नहीं दिया होगा कि distance between two base pair is this much, then also you put this value. So you have to remember this value. You have to keep in your mind this value, the distance between two base pair is 0.34 nanometer. ये जो चीज है, ये जो डेटा है, ये जो वैल्यू है, आपको याद रखना पड़ेगा. अगर क्वेश्चन में दिया होगा तो बहुत अच्छी बात है, अगर नहीं दिया होगा तो वहां पे यू हैव टू पुट दिस वैल्यू. Okay, so now if the length of the equally DNA is this much and you know the distance between two consecutive nucleotide, okay, or else you can say the distance between two, what is the distance between two base pair? Is the same thing. If I'm saying the distance between two base pair, what does it mean? It means the distance between two consecutive nucleotide, right? Okay, so here the value is what? 0.34 nanometer. So what we need to do, we need to find out the number of base pair. How many base pair are there? So you can see the number of base pair in E. coli is equal to 1.36 millimeter divided by 0.34 nanometer. Just remember one thing, you have already read about it in your lower grade. हमेशा यूनिट को सेम यूनिट में लाना है. यहाँ पे ये मिलीमीटर में है, ये नैनोमीटर में है. 1 millimeter is equal to how many meter? 1 millimeter is equal to 1 into 10 to the power minus 3 meter. Okay, and if I'm saying 0.34 nanometer, अगर मैं नैनोमीटर को मीटर में कन्वर्ट करूं, यहाँ पे कितना होगा वैल्यू? 0.34 into 10 to the power minus 9. तो हमें ये जो दो यूनिट है इसे मीटर में लाना है. अलग अलग यूनिट में हम क्वेश्चन सॉल्व नहीं कर सकते. तो ये जो यूनिट है इसे हमें सेम यूनिट में लाना है, इसे हमें मीटर में लाना है. तो 1.36 millimeter and here it is 0.34 nanometer. So 1.36 into 10 to the power minus 3 divided by 0.34 into 10 to the power minus 9. So the answer will be this. Okay, answer will be this. This is your NCERT textbook question. I hope it is clear. I hope it is clear. You just need to remember what is the distance between two nucleotide, or else you can say what is the distance between two base pair, this and this. I'm talking about this distance. What is the distance between two base pair? Okay, and how many, if someone will ask you about the pitch, you need to remember how many base pair are there in one turn. If someone will ask you about the pitch, then you need to remember how many base pair are there in one turn. Pitch means they are asking about the length of one turn. Okay, don't get confused. Pitch means they are asking about the length of one turn. Now the question is how many base pair are there in one turn? So the total number of base pair in one turn is 10. You have to remember that. It is approximately 10. In your higher class, you are going to read that there is some variation. But for now, just remember the total number of base pair in one turn is 10. And the distance between two nitrogenous bases, or else you can say the distance between two base pair, not two nitrogenous bases, the distance between two base pair. Base pair means two nitrogenous bases, they are attached to each other. They are paired with each other. That's why they are called base pair. If there is only A, if there is only T, if there is only G, if there is only C, those are nitrogenous bases. Those are not base pair. When they are pairing with each other, when A is paired with T and G is paired with C, then they are called base pair. Okay, you have to be clear about this term. So, I hope this question is clear. Now, let us move to the next part. Group the following as nitrogenous bases and nucleoside. Group the following as nitrogenous bases and nucleoside. Now, the question is, what is nucleoside? Nucleoside means the nitrogenous bases connected to pentose sugar. Nitrogenous base connected to pentose sugar by glycosidic bond. That structure is called nucleoside. What is nucleoside? Nitrogenous bases. Nitrogenous base, there is only one base, not bases, nitrogenous base connected to pentose sugar by glycosidic bond. And what is nucleotide? Nucleotide means nitrogenous base connected to pentose sugar and there is a phosphate. So or else you can say nucleoside plus phosphate is called nucleotide. Nucleoside plus phosphate is called nucleotide. What is nucleoside? Nitrogenous bases connected to pentose sugar by glycosidic bond. We have already read about it in our previous lecture. So group the following as nitrogenous bases and nucleoside. You can see adenine, cytidine, thymine, guanosine, uracil and cytosine. Just remember one thing, you need to remember about the nitrogenous bases. Okay, so nitrogenous bases are broadly classified into two types, purine and pyrimidine. Under purine, adenine and guanine is coming. Under pyrimidine, what are coming? Cytosine, uracil and thymine. So as you can see, yeah, so this is what adenine. Adenine is a nitrogenous base. This thymine is also a nitrogenous base. Uracil is also a nitrogenous base. Cytosine is also a nitrogenous base. The rest are what? The rest are, we have already read about it. You just need to remember what are the nitrogenous bases: adenine, guanine, cytosine, uracil, and thiamine. Okay, so guanosine is not the nitrogenous base. Guanine is the nitrogenous base. Guanosine is, it is a nucleoside. Cytidine is a nucleoside. So these are what? These are nucleoside. Cytidine and guanosine. These are nucleoside. So all those things that I have marked in yellow, they are nitrogenous bases, and all those things that are marked in red, they are nucleoside. Okay, you just need to remember what are the nitrogenous bases. Okay, if there is any slight variation in the name, then they will come under nucleoside or nucleotide. If a double-stranded DNA has 20% of cytosine, calculate the percentage of adenine in the DNA. So you can solve this question on the basis of Chargaff rule. I think all of you know what is the Chargaff rule. In case you don't remember, you can simply solve this question by remembering just one thing, that A will always pair with T and G will always pair with C. So the number of A and the number of T will be same, and number of G and number of C will be same in a double-stranded DNA. You can see A will always pair with T and G will always pair with C. So if it is given that in a double-stranded DNA, the amount of cytosine is 20%, then the amount of guanine will be, it will be the same. It will be the 20% only. It will be the same. And if it is given that the amount of adenine present in DNA is 30, then the value of thiamine will also be 30. So let us solve this question. If a double-stranded DNA has 20% of cytosine, you know that cytosine always pair with guanine. So if the value of cytosine is given, if the amount of cytosine is given, here the cytosine is 20%, so guanine will be, C is 20%. So G will be the same, 20%. We don't know the value of A, but we know that A will be similar to, A will be equal to T, because A always pair with T. We don't know the value of A, but we know that A का value जितना रहेगा, T का value उतना रहेगा. So suppose I'm taking A as X. What will be the value of T? The amount of T will be the same. It will be the X only. And total percentage we are taking 100. So what we need to do? C plus G plus A plus T is equal to 100. So if C and G is 20 and 20, so 40 plus X is how much? 2X. That is equal to 100. We can easily find the value of X now. What will be the value of X here? 2X is equal to 60. And X is equal to how much? X is equal to 30. Right? So the percentage of adenine in the DNA is how much? The percentage of adenine is 30. What will be the percentage of thiamine? If the percent of adenine is 30, what will be the percent of thiamine? It will be the same. It will be 30 because adenine always pair with thiamine. And the percentage of cytosine is also given. It is 20%. Then what will be the percentage of guanine? It will be 20%. Okay. So here the answer will be 30. So I hope this is clear, whatever we have discussed till now. So calculate the percentage of adenine in the DNA. The percentage is 30%. And if someone will ask you what is the percentage of thiamine, it will be the same. It will be the same 30%. And here the cytosine, the percentage of cytosine is given, that is 20%. If someone will ask you what is the percentage of guanine, it will be the same, 20% only. I hope it is clear. Yeah. So now just have a look. This is another question. Again, it is a textbook question only. If the sequence of one strand of DNA is written as this, write down the sequence of complementary strand in the 5' to 3' direction. When I'm saying complementary strand, means they are complement. They are doing what? They are complementing each other. Okay. So if you can recall the double helical structure of DNA, there are two strands. And the polarity, if I'm talking about the polarity, the two strands, they are anti-parallel to each other. If the polarity of one strand is 5' to 3', then the polarity of other strand will be 3' to 5'. Okay, so and you also know one more thing, that A always pair with T and G always pair with C. On that basis, we can easily find out what will be the sequence of the complementary strand in the 5 prime 3 prime direction. So you can see here, if the sequence of one strand of DNA is written as this, write down the sequence of complementary strand in the opposite direction. It is 3 prime to 5 prime direction. Here it will be 3 prime to 5 prime. So you can see A will always pair with what? A will always pair with T, right? And T will always pair with A. G will always pair with C. C will always pair with G. Okay, A will always pair with T. T will always pair with A. A will always pair with, then G will always pair with C, and C will always pair with G. And what will be the polarity? 3 prime. Here it will be 5 prime, as they are anti-parallel to each other. They are not parallel, they are anti-parallel. When I am saying parallel, means they are in same direction. Both the line, they are in same direction. When I am saying anti-parallel, the direction will be opposite. Direction will be opposite. Okay, so the polarity of one strand, if the polarity of one strand is 5 prime to 3 prime, then the polarity of the complementary strand will be 3 prime to 5 prime. Okay, and you also know that A always pair with T and G always pair with C. On that basis, you can easily solve this question. Okay, I hope it is clear. Now, let us move to the next question. This is a PYQs. This is a previous year question, AIMS 2017. ओके, ये एम्स 2017 में आया था, ये क्वेश्चन असर्शन रीजन वाला क्वेश्चन आया था एम्स 2017 में. तो इसमें क्या पूछा गया है? इन अ डीएनए मॉलिक्यूल एटी रीच पार्ट मेल्ट बिफोर जीसी रीच पार्ट. यस, why so? Because we know that A pairs with T. How many hydrogen bonds are there? There are 2 hydrogen bonds between A and T. And if I'm talking about GC, okay, G pairs with C. How many hydrogen bonds are there between GC? 3 hydrogen bonds are there. So if someone will ask you that I want to break the double-stranded DNA, so from what part I should start breaking? You can see the compatibility between A and T is less. There are only 2 hydrogen bonds. But the compatibility between G and C is more because there are 3 hydrogen bonds. So it will be easier for us to break 2 hydrogen bond in comparison to 3 hydrogen bond. Okay, so यहाँ पे लिखा गया है in a DNA molecule 80 reach parts melt before GC reach part. Yes, it is correct because 80 reach part जो है वहां पे 2 हाइड्रोजन बॉन्ड है जैसे हम इजीली तोड़ सकते हैं. नो डाउट एनर्जी लगेगा बट इजीली तोड़ सकते हैं इन कंपैरिजन टू जी एंड सी, बिकॉज जी एंड सी के बीच जो हाइड्रोजन बॉन्ड है 3 हाइड्रोजन बॉन्ड है. द रीजन इज in between A and T there are 3 hydrogen bond, whereas in between G and C there are 2 hydrogen bond. तो यहाँ पे ये लोग गलत स्टेटमेंट दे दिए. तो पहला वाला जो स्टेटमेंट है जो वो असर्शन में डाले हैं वो सही है, बट जो दूसरा वाला स्टेटमेंट है जो रीजन में वो लोग डाले हैं वो गलत है. क्यों? क्योंकि यहाँ पे लिखा गया है कि इन बिटवीन ए एंड टी देर आर 3 हाइड्रोजन बॉन्ड. ए एंड टी के बीच 3 हाइड्रोजन बॉन्ड नहीं होता है. ए एंड टी के बीच 2 हाइड्रोजन बॉन्ड होता है एंड जी एंड सी के बीच 3 हाइड्रोजन बॉन्ड होता है. Okay, so this one is wrong. This one is wrong. So the reason is wrong and the assertion is correct. So assertion is true and reason is false. Okay, assertion is true and reason is false. Okay, so I hope it is clear. This is a PYQ. I'm repeating. It's a previous year question. किसमें आया था? एम्स में आया था. एम्स 2017 में ये क्वेश्चन आया था. देखो कितना इजी है. यू जस्ट नीड टू रिमेंबर ए और टी के बीच कितना हाइड्रोजन बॉन्ड है, जी और सी के बीच कितना हाइड्रोजन बॉन्ड है. उसी के बेसिस पे हम पूरा क्वेश्चन सॉल्व कर सकते हैं. ओके, नाउ लेट अस मूव टू द नेक्स्ट पार्ट. Nucleotides are building block of nucleic acid. Each nucleotide is a composite molecule formed by, हम लोग ऑलरेडी प्रीवियस क्लास में पढ़ चुके हैं न्यूक्लियोटाइड क्या है, न्यूक्लियोसाइड क्या है. सो न्यूक्लियोसाइड क्या है? न्यूक्लियोसाइड क्या है? न्यूक्लियोसाइड मीन्स यू जस्ट नीड टू कीप इन योर माइंड नाइट्रोजनस बेस प्लस पेंटोज सुगर. हाउ दे आर कनेक्टेड विथ ईच अदर? बाय ग्लाइकोसाइडिक बॉन्ड. Okay, and if I am talking about nucleotide, nitrogenous base plus pentose sugar plus phosphate. Okay, when phosphate group is added to nucleoside it is called nucleotide. यहाँ पे बच्चे बहुत कंफ्यूज होते हैं, तो बस इतना है. न्यूक्लियोसाइड क्या है? न्यूक्लियोसाइड है नाइट्रोजनस बेस एंड पेंटोज शुगर, ओके। एंड उसमें अगर मैं एक फॉस्फेट ग्रुप ऐड कर देती हूँ तो वो बन जाएगा न्यूक्लियोटाइड, ओके। पहले आएगा न्यूक्लियोसाइड, उसमें फॉस्फेट ग्रुप ऐड कर दो, वो बन जाएगा न्यूक्लियोटाइड, ओके। सो न्यूक्लियोटाइड are building block of nucleic acid. Each nucleotide is a composite molecule formed by, यहाँ पे हम देखते हैं कि हम इस क्वेश्चन को कैसे सॉल्व करेंगे, ओके। कहाँ पे कंफ्यूजन होने वाला है, कहाँ पे कंफ्यूजन होने वाला है, शुगर प्लस फॉस्फेट, ये पूरा हम काट सकते हैं, यहाँ पे कंफ्यूजन होना ही नहीं है, ओके। शुगर प्लस फॉस्फेट ये पूरा काट सकते हैं, शुगर बेस एंड OH नहीं, ये भी नहीं होगा। तो ये 2 पॉइंट पे हम कंफ्यूज हो सकते हैं। पर अगर ध्यान से देखा जाए तो यहाँ पे पूछा गया है ईच न्यूक्लियोटाइड, एक न्यूक्लियोटाइड के बारे में वो लोग पूछे हैं। तो एक न्यूक्लियोटाइड में क्या क्या है, बेस है, शुगर है, फॉस्फेट है। पॉलीन्यूक्लियोटाइड नहीं बोले हैं, अगर पॉलीन्यूक्लियोटाइड बोलते या बोलते तो स्ट्रक्चर ऑफ डीएनए के बारे में पूछते तो स्ट्रक्चर ये होता। बट यहाँ पे वो लोग खाली न्यूक्लियोटाइड के बारे में पूछे हैं, ईच न्यूक्लियोटाइड। प्लीज रीड द क्वेश्चन केयरफुली, क्योंकि एम्स में जो क्वेश्चन आता है वो क्वेश्चन बहुत इजी होता है, लेकिन वो आपको बातों में घुमाते हैं, वहां पे थोड़ा सा ट्रिक करते हैं। तो बच्चे क्या बोलेंगे, न्यूक्लियोटाइड्स, यहाँ पे तो न्यूक्लियोटाइड्स लिखा है, तो चलो डाल देते हैं पॉलीन्यूक्लियोटाइड सोच के ये वाला लगा के आ जाएंगे, बट आंसर होगा ये बेस, शुगर एंड फॉस्फेट। ईच न्यूक्लियोटाइड, यहाँ पे देखो क्वेश्चन, ईच न्यूक्लियोटाइड इज ए कंपोजिट मॉलिक्यूल फॉर्म्ड बाय बेस, शुगर एंड फॉस्फेट। यहाँ पे एन नहीं होगा, एन मतलब एन इज इक्वल टू 1 2 3 एनीथिंग, एनीथिंग इज पॉसिबल, बट यहाँ पे एन नहीं होगा। ये वाला जो आंसर है ये किसके लिए होगा, पॉलीन्यूक्लियोटाइड के लिए होगा, ओके। So answer will be A. I hope it is clear. Yeah. So in our next class, we are going to study about DNA replication. So these are the things that you are going to study in your next class. What do you mean by replication? What are the steps in replication? What are the enzymes involved? What are the raw materials required? What is semi-conservative method of replication? Okay, semi-conservative method क्या है? रेप्लिकेशन के लिए हमें क्या क्या रॉ मटेरियल्स चाहिए, जैसे जैसे कुछ खाना बनाने के लिए हमें इंग्रेडिएंट्स चाहिए, ठीक उसी तरह रेप्लिकेशन के लिए भी हमें कुछ कुछ चीजें चाहिए, कुछ कुछ टूल्स चाहिए, कुछ एंजाइम्स चाहिए। सो व्हाट आर द रॉ मटेरियल्स रिक्वायर्ड, व्हाट आर द एंजाइम्स इन्वॉल्व, व्हाट आर द स्टेप्स इन रेप्लिकेशन, what do you mean by replication? So these are the things we are going to study in our next class, okay. I hope you all enjoy the session. I hope you all enjoyed this session. So this is all about today. If you have any doubt related to this question, you can drop your queries in the chat box. Okay. So all of you, please take care. And if you like my session, please do like, share and subscribe. And please go through the previous lecture because it will be very much easier for you to solve this question once you go through it. This much for today. Take care. Bye everyone.